Hello students, welcome back. This is module 4.1.1, Introduction to DNA. This is the first module of Unit 4. Earlier in the semester we studied biological macromolecules, and at that time, of course, we studied proteins, carbohydrates, lipids, and nucleic acids. Nucleic acids were the fourth category of biological macromolecule, and we really barely touched on them at that time. And I told you we'd be back, and here we are. We're back, baby. Okay. Let's talk about some of the major functions of nucleic acids. Remarkably, these are chemicals which have the ability to store information. It really makes them a unique type of chemical. They also function in energy transfer, think of ATP and that kind of thing. And some very few have what is otherwise exclusively limited to proteins. Some very few have that ability to serve as enzymes. So that would be mostly RNA. Here's some examples. DNA, RNA, and ATP are among the most important and prominent nucleic acids. So we have studied inheritance in our last unit, Mendelian genetics and such, and now we're going to continue and look in more depth at DNA and how it functions, because DNA is, of course, what chromosomes and genes are made of. The material that we're going to be talking about here, this falls under the category of molecular genetics. And so we can talk about DNA. In the past, I told you it was enough that you just simply know DNA. Now you should absolutely know that DNA stands for deoxyribonucleic acid. And DNA was discovered in 1869 by Friedrich Mischler, a German chemist. He called it at that time nuclein because he knew it was associated with the nucleus, but that was about all he knew. The function was unknown then and remained unknown for decades into the future. We know now that DNA contains the genetic code, the information of life, the blueprint for an organism, talking in kind of making an analogy, I guess. In any case, this is a molecule that's capable of storing information, of reading that information, and of copying that information. That is just a remarkable feat for a chemical. Much of life is controlled by DNA, and much of modern biology revolves around understanding and manipulating DNA. So if we look back at the history of the discovery of DNA and its significance, early on, before biologists really knew what was going on, they understood there had to be some way that life was passed on, the characteristics of life were passed on from parent to offspring. And in the life cycle of every organism, there was one point where it consisted of a single cell. And so they knew that the secret of life, the genetic material, had to be present inside a single cell. And so they started to look at different areas of the cell, and they removed various organelles and whatnot, and several early experiments concluded that the genetic information had to be found in the nucleus of the cell, as cells with the nuclei removed could not divide and they didn't function normally. And so they knew that that important material must have been in the nucleus. And just to remind you, because we are, of course, bacteria are also alive, but prokaryotes have DNA, but it's just in a region. It's just not inside a nuclear membrane. And so when we talk about genetic information, that is the hereditary information. And of course, then once we understood that the genetic information resided in the nucleus, then we want to know what is this genetic material. And so lots of early experiments were performed trying to figure this out. Now, when we analyzed the content of the nucleus, we found out that chromosomes, which filled much of the nucleus, were made of 60% protein and 40% DNA by weight or by mass. And they had to figure out which of these two major constituents of the nucleus were the genetic material and contained the genes. Initially, protein was favored. It was seen as being very complex. Protein has 20 different amino acid monomers that make it up. And DNA, by contrast, was perceived as being very simple. DNA has only 4 monomers that make it up. These are nucleotides. And as a consequence, many people favored protein. And so one of the very important early experiments that was done by Alfred Hershey and Martha Chase. And really the point of their experiment is right here, is protein or DNA, the genetic material. Now, their system that they used to explore this question, it had 2 major components. One of the components was bacteriophages. These are viruses that attack bacteria. So a bacteriophage is simply, it will, when it attacks, well, I'll describe the attack a little bit later. But let's define for you what viruses are. A virus is an obligate intracellular parasite, and they consist of a nucleic acid surrounded by a protein coat. And whether or not viruses are alive, well, according to the cell theory, they certainly are not. But because viruses, when we say they're obligate intracellular parasites, it means they have to live inside cells. If there are no cells, viruses don't do anything. They sit there like a bump. And when a virus attacks, it attaches to the cell that it's attacking, and it injects its genetic material into the cell, where that genetic material takes over the DNA of that cell. So if you wonder why we call it a computer virus, well, that's kind of what a computer virus does, except a real virus goes a little bit further. It not only takes over the information, but it uses that information to make more viruses. And eventually the host cell, the original host cell, may end up splitting and releasing many more viral particles that go out and infect other cells. So kind of an interesting thing. So really the reason that they wanted to use bacteriophages, these viruses, was because they figured whichever was injected, if it was protein or DNA, that's excuse me, what allowed viruses to take over the cell. And so that's why they were trying to figure this out. Now, how are they going to figure out which chemical is injected? Well, they need a second part to their experimental setup, and they used radioisotopes, or isotopes that are radioactive. And so they labeled 2 molecules that are found in viruses. Obviously, one's going to be in protein primarily, and one's going to be in DNA primarily. And you want a molecule that's mostly in DNA, but not in protein, and one that's mostly in protein and not in DNA. And so they labeled the protein sulfur in the protein. So sulfur is very common in protein, and there's no protein whatsoever in DNA. They labeled the DNA phosphorus molecules, pardon me. And so there is a tiny, tiny bit of phosphorus in protein at times, but there's a lot of phosphorus in DNA. And so these are considered good molecules to distinctively label DNA and protein. And so then they grew their bacteriophages, the viruses, in separate colonies. And one of the colonies had bacteria that had the hot sulfur, and the other colony had phosphorus that was radioactive. And so they could follow this. And so it ended up that the viruses themselves were then labeled with either hot sulfur or hot phosphorus in the 2 separate trials. In one trial, the protein was labeled, and the other, the DNA, was labeled. And so here we can see the one in which the sulfur was labeled. This little thing that looks like the lunar lander, that represents a virus, and it's on this ovoid shape, which is the bacterium, and it injects its genetic material. And now we know that what was injected was DNA, but at that time they didn't know. And the labeled sulfur stayed outside, and they could actually use a centrifuge which separates mass, and they could figure out which was inside the bacterium. And later they let the bacteria go on and grow, and so they could tell that the genetic material of the virus had been successfully injected. And so the first trial showed that the protein was not the genetic material, and so from that we knew that it was not the protein. And why do we have to do a second trial then? We have eliminated the protein as a possible candidate for the genetic material. Well, the reason we had to do a second one was because it could have been something else besides the protein, and it or besides the DNA. The first trial showed it couldn't be the protein. The second trial showed that it was DNA. And here we can see what happened in the second trial. The genetic material was injected, and they found out that indeed by measuring it with what they call a scintillation counter, a little kind of like a Geiger counter, only for very precise measurements, they could tell that it was inside the cell, and those cells later went on and divided. And so we know that the material, the genetic material, was DNA after Hershey and Chase's experiment. Here is just a graphic showing what the bacteriophage looks like. It was actually called the T4 bacteriophage that they use. Here's some actual photomicrographs. These are tiny, and this is remarkable pictures because it actually shows the DNA being injected into this brownish bacterial cell here at the bottom. And so Hershey and Chase, their results indicated that DNA was the genetic material. And after that, the race was on to discover the structure and function of DNA. And so we'll continue there with our next module on what was the structure of DNA.